So the most likely sequence alignment between these two sequences are the one that is indicated here, taking into account a mismatch here, a match here, a gap here, etc. So the idea is here to basically cut a big problem, that is the alignment of the whole sequences, into smaller problems and then compute the alignment. So the Smith-Waterman alignment is a kind of local alignment that determines similar regions between two strings of nucleotide. It can also be used for proteins. But instead of looking at the total sequence, this algorithm is comparing segments of all possible lengths. And here we don't have the scoring that is negative. The main difference with the previous one is that the score starts at 0. So we then backtrack. If you follow here the blue arrows or the blue numbers, so the numbers are affected to, these are the scores of each position, and you backtrack following the scores that you have here until a cell with a 0 score is reached. And this yields the highest local alignment. So we won't be going into more details, but I just wanted you to have an idea about these two main algorithms for respectively global and local alignments. So the word-based methods now for pairwise alignment are methods for which an optimal alignment is not guaranteed, but efficient and faster, more efficient and faster than dynamic programming. So it is useful for databases searches, and in fact this is an approach that we call heuristic approach. I'm giving you here the example of the BLAST, that is a heuristic word-based method. So heuristic method is basically any approach of problem solving that would be that would not guarantee an optimal or perfect alignment, but that would be sufficient for your goal. So the idea is to cut the sequence into small portions or words of a specific length that is called K, and then use it to screen a database. So these methods are especially useful when you have large-scale database searches, where it is expected that you will have a whole bunch of candidate sequences that will essentially have no significant match to your query. So this is the example of the BLAST, which is stands for Basic Local Alignment Search Tool. It is an algorithm to compare a query sequence to a library or database of sequences. It allows you to estimate the identity with a certain confidence threshold. And it is really popular in the scientific community. BLAST is maybe one of the most popular ones. And it is used, for example, if you want to know what species or organisms have a particular genes you are interested in other than the one that you are extracting the sequence from. So in the BLAST method, you have the query sequence here. You basically can first use an optional first step that is filtering the low complexity regions in order to exclude them from the next steps of the analysis. Then you do what we call a seeding step, which is listing all possible words of length K, which here is 11 by default for the DNA. If you were comparing protein sequences, it would be 3 by default. So here it's 11. So all possible words of length 11 are written here. Then you search the database for matching words using a scoring matrix, as we saw it. So you calculate that matching score, and you evaluate a threshold score in order to be able to top rank the most similar sequences. Then the process is repeated for all words of the query. And in fact, once you have identified, so this is your query and this is the sequence in the database. So once you identified a match, what you do is perform an extension on both ends only if the alignment is above the threshold that is fixed. And the contiguous alignment without gaps would allow you to identify a potential score, and the highest possible score, which would be called the HSP, the high scoring segment pair, would be the most highly confidently aligned sequence to your query. So, as I told you, a scoring matrix is used to evaluate the quality of the alignment. So basically it would be defined as a substitution matrix that is predefined. So you decide of what scores you would be giving to a match, or to a mismatch, or to a gap. And blossom matrices are an example of this kind of scoring matrix. I'm sure you would have encountered this before. I won't have time to cover all these kind of matrices. So PAM is another one, but blossom might be also really useful. And this allows you to judge the quality of your alignment. So these are scoring for each kind of changes that you would have in between the two sequences compared that you are comparing. So this is the classical web interface for a BLAST search. So I used it for one particular query. As I told you, if you remember, you can query using a FASTA sequence. You can enter an accession number like the GI. So I used here 1. And I'm highlighting here just a very basic alignment using this database. Now I would like to highlight something that you can access here through the algorithm parameters. That would be what you are seeing here. So these are the parameters I've been talking to you about. So you can decide of the word size, and you can also decide of the scoring parameters, and the match and mismatch scores are indicated here. And the gap costs could be linear or could be predefined, and you can choose from one of them depending on what value you would like to affect to a gap opening or to a gap extension. So this is the result output. So an output in a BLAST search would basically be a different variety of formats. You can have HTML, you can have plain text, you can have XML. For NCBI, by default, you would have an HTML. So this is an example of this kind of output. First section would be a list of the sequences that have the best match. So this is your query, and these are the different matches. A second information is given in this table, where you have the matching sequences and several kind of metrics, among which you would find the e-value, which is the probability that the alignment is found by chance. So this takes into account the number of nucleotides in your reference, the number of nucleotides in the query, and the probability that the alignment would be made by chance. So the lower the e-value, the more interesting is the match. And another section in this output is given by the third section, which gives you alignment details. So these are basically your query and your subject in the database, and percentage of identities between the sequences, the number of gaps, etc. So you have different variant queries according to the type of query sequence and the type of sequence in the database. So basically what we used here for nucleotide search is the BLAST N, but you can have all different kind of variants, and we also have a mega blast or psi blast options. There are a lot of them that you can retrieve through that web page. So multiple sequence alignment now. So we just saw the pairwise alignment. The multiple sequence alignment now have been developed in order to handle more than two sequences at a time. it aligns the queried sequences in order to form a query group. So in fact, here you can identify conserved sequences and homology that can be inferred, and you also can know the evolutionary relationship between the sequences that are studied. So different methods again here, dynamic programming or progressive methods, or HMM. That would all allow you to evaluate the conservation across your sequences. So the dynamic programming is generally not really optimal for more than 2, 3, or 4 sequences, so it would be here computationally expensive, and it basically extends the sequence matrix from 2 sequences to the number of sequences in the query. So it would be basically aligning different pairs of sequences and constructing n times the sequence matrix formed from each 2 sequences until the whole number of sequences in the query is analyzed. An example of this is the MSA. You have also progressive methods that would allow you to align sequences in order to identify the most similar one, and they progressively add all other related sequences to the group. So let's see the example of Clustal in order to highlight this. So there are different progressive methods, of course. I will only be talking about the Clustal here. So Clustal basically takes the sequences that you have as an input and computes the pairwise alignment and distance matrix. So you have all possible pairwise alignments of the query sequences, and then it calculates a similarity score between all these possible pairs using that distance matrix that we've been talking about. And these scores would be used to build what we call a guide tree. And this guide tree would be used as a template to progressively align all the sequences that you have in your query. And this continues for any additional data or any additional sequence that you have until you finish all the sequences that you have in your query. And in fact, what you do here is that you add gaps during the alignment in order to optimize the sequence alignment after you constructed your guide tree. So HMMs are a third type of multiple sequence alignment process. It determines the probability score for multiple sequence alignments, and the aligned sequences would serve as a group, so there is no need to previously order the sequences. And you use here what we call a Viterbi algorithm to determine the most likely path that could have produced the sequence, taking into account insert state, match state, or deletion state. And it is used to build what we call an HMM profile, and it's improved for more distant sequences. It is widely used for proteins as well as for DNAs, and it's really highly popular because it is really robust. Now we will be talking about the conservation scores that are related to the multiple sequence alignment in the next video.